Do you remember when you first learned about DNA? Because I sure do. It felt like falling in love for the very first time. And like falling in love, it changed the course of my life. It's changing the course of your life too, whether you know it or not. See, maybe you're like me. I was coming from software engineering as a background, and biology seemed like this arbitrary, messy study. I didn't get it. I thought it was about looking at things under microscopes, memorizing arcane Latin terms, and dissecting animals. But then I heard about DNA, and I came to understand that underlying that messy diversity of living systems is a single, elegant code, the code of life. And I just had to learn it. So I went off to graduate school just before the turn of the century to study genetics, and I was appalled at the tools that were available to read DNA. They were slow, painstaking, very manual, not to mention radioactive. Such a disconnect between this incredibly exciting, complex code and our ability to even read it, much less understand it. Fortunately, during that time, there was a whole spate of innovations that really drove our ability to read DNA. These are things like tagging the individual bases, A's, T's, G's, and C's, with individual fluorescent labels so that we could fire lasers at them and read them moving through microfluidics. So really automate and rapidly read the DNA code. This all culminated in the year 2000, the declaration that we had cracked the code, sequenced the full human genome, all 3 billion base pairs. Now, to put this in context, 3 billion bases is one of the larger code bases out there. So as significant as a milestone as this is, in order to understand the code, we're certainly going to need to read more than just one copy of it. Fortunately, another decade of innovations in the tool chain, the hardware, wetware, and software came along, such that we could really speed up the reading of individual human genomes. This led to a precipitous drop in the cost of sequencing genomes, even faster than Moore's law, going from the billions of dollars it took to read the first human genome to only a few $100s to read a whole human genome. This puts us in sight of being able to sequence the whole human genome of every one of the billions of humans on this planet. It also really turns biology into an information science, which is incredibly exciting this day and age because it means we can bring the tools of information science, including AI, to bear to understanding that code. But what would it take to make it a programming science or an engineering discipline? Well, we're going to need to be able to write that code as well. Today, the tools for writing DNA as code are still fairly limited. We can only write short snippets of DNA at a time. Then we have to paste them all together to form code we'd like to deploy. And then we have to get that into the operating system, that is, the cell where the human genome resides. Our choices today for deploying code in cells include things like shocking it in, squirting it in using viruses, or even firing it in on microscopic gold bullets. It's kind of barbaric. Also, much of the time, it doesn't work the way you'd expect it to. The human genome is not a deterministic programming environment. It can do things that you didn't expect. It's not predictable. And sometimes it just doesn't work at all. So we still have a ways to go before the human genome is programmable. But in the meantime, we've had some great parallel innovations that are changing the way we think about the human genome. One is the discovery of enzymes like CRISPR, which quite rightly received the Nobel Prize in 2020. They don't write DNA from scratch, but they do allow us to go in and change bases in the human genome quite precisely. Last year, a watershed moment. Scientists announced that they had, for the very first time, used these kinds of editing enzymes to change the genome of a living human, saving the life of a baby who otherwise would have had a fatal genetic disease. In parallel, we're learning that the DNA in our cells is not the only code that matters in the human genome. In fact, there are tiny particles floating throughout our body called extracellular vesicles or exosomes that also carry snippets of code between all of the cells, executing their commands and communicating with each other. If cells are the operating system, you can think of exosomes as the network. So it stands to reason that there could be orders of magnitude more information there than even in the cells themselves, and we can use the modern tools of DNA sequencing to find out what's going on in there. Not only that, we can isolate those exosomes, load them up with snippets of our own code, and actually deploy them to precise places in the body as little containers that can execute code for us. This is really transforming how we think about the power of the human genome for both diagnostics and therapeutics. So, a quarter of the century in, we're still at the hello world stage of programming the human genome, but we can't wait to see what comes next.